The lecture is now talking about drug receptors and pharmacodynamic. As we had learned before that the kinetic is the effect of the body on the drug, and we learned there many terms such as the absorption, volume of distribution, metabolism, and other terms. Now we will go through the drug receptor and pharmacodynamics. That means the effect of drug on the body. So pharmacology, what is drug does to the body, drug action and effect. As we have said before, the effect of drugs on the body is the actually pharmacodynamic, and the effect of the body on the drug is the pharmacokinetic, what body does to the metabolism. Now pharmacodynamic phase, process of drug action that occur when drug reaches site of action and produces an effect. This is the most important two terms in this lecture. What is action? What is effect? When we say what is the action of this drug, it means how the drug works, usually by enhancing or inhibiting cell functions. It means all the action of the drug, whether it is desired or not. While the effect is the consequences of drug action on the body. That means what will happen after the drug binds to its site of action. After its binding, there is an event happens. This is called the effect. Well, the major example here, aspirin, the action actually block prostaglandin synthesis, while its effect is analgesia and antipyresis. That means when I ask you in the exam, give me the action of aspirin, you have to say that actually it blocks prostaglandin synthesis and whatever mechanism of action. While when I tell you, give me the pharmacological effect of aspirin, at that time you will say it's analgesia and antipyresis. Now, how drugs act. Many drugs exert their action by binding to protein target on mammalian cells. This protein or these proteins actually are receptors, ion channels, enzymes, carrier molecules. However, there are exceptions such as the antibiotics, for example, and anti-cancer drugs, etc. So the majority of drugs act on either receptors, ion channels, enzyme, carrier molecule. Well, let's go through receptors. What is receptors? Receptors are a specific or the specific molecule in a biologic system with which drugs interact to produce changes in the function of the system. Receptors must be selective in their ligand binding characteristics so as to respond to the proper chemical signals and not to meaningless ones. Receptor must also be modifiable when they bind to a drug molecule so as to bring about the functional changes. Moreover, receptors largely determine the quantitative relations between dose or concentration of a drug and pharmacologic effect. Well, let me explain this to you. That means when there is a large amount of receptors, what can we expect in regarding to response? It means maybe this lead to increase the response or the effect. Why? Because large amount of drug bind to large amount of receptor in comparison to the action happened due to the existence of low amount of receptor. At that time, I can expect that the effect is less than that with high amount of receptors. Also, receptors are responsible for selectivity of drug action. Receptor mediate the action of both pharmacologic agonist and antagonist. What does agonist do? Actually, agonists activate the receptor to signal as direct results of binding to it, while antagonists bind to receptor but do not activate or generate any signal. Well, the figure here shows you how the substrate, when it binds to a receptor or an enzyme or whatever, at that time in a theory called lock and key, when it is mimic the figure or it's like the figure of receptor, once it binds it, make a complex, and this complex alleviate or initiate a signal which is responsible on the action after that. Now the effectors are molecules that translate the drug-receptor interaction into a change in cellular activity. The best example of effectors are enzymes such as adenylyl cyclase. Some receptors are also effectors in that a single molecule may incorporate both the drug binding site and the effector mechanism. For example, tyrosine kinase effector is part of the insulin receptor molecule, and a sodium-potassium channel is the effector part of the nicotine acetylcholine receptor. That means when insulin binds to its receptor, tyrosine kinase is stimulated. When acetylcholine binds to its receptor, sodium-potassium channel actually is affected. This is what means an effector. Now, graded dose response relationship. When the response of a receptor effector system is measured against increasing concentration of a drug, the graph of the response versus the drug concentration or dose is called a graded dose response curve. Well, at night when I will explain the lecture again to you by Zoom, I will show you the curve how it be, and the efficacy, the Emax or the maximum effect, and potency, the EC50 or ED50 parameters are derived from these data. The smaller the EC50 or ED50, the greater the potency of the drug. See here this figure. We draw the dose versus the change in heart rate. Well, you can see here at the peak is the Emax, while the EC50 or the ED50 here is maybe 5. You can see here 5 milligram or whatever, while Emax you can see too high. This is the differences, and at night I will explain to you more. Well, there is same data logarithmic dose axis. The dose or concentrations at which effect is half maximal is denoted as EC50, whereas the maximal effect is the ED50. This curve called logarithmic dose. Well, graded dose binding relationships and binding affinity. If the percentage of receptors that bind drug is plotted against drug concentration, a similar curve is obtained. Actually, the concentration at which 50% from the receptors are bounded is denoted as KD, and the maximal number of receptors bound is termed as Bmax. This graph explains what I had said now, what I have just said now. You can see here the KD and the Bmax. The smaller the KD, the greater the affinity of the drug for its receptor. If the number of binding sites on each receptor molecule is known, it's possible to determine the total number of receptors in the system for the Bmax drug-receptor binding. That means drug plus receptor equals drug-receptor complex lead to biologic effect. Moreover, the quantal dose response relationship is defined as the minimum dose required to produce a specific response is determined in each member of a population. Quantal dose response plots from a study of a therapeutic and lethal effect of a new drug in mice. The median effective and median toxic, and in animals median lethal dose are derived from the experiment carried out in the manner. Actually, my students, I will teach you this in the laboratory, inshallah, when we will take the toxicity. Here, from the quantal dose response, the minimum dose required to produce a specific response. Here, this figure showed the cumulative percent exhibiting therapeutic effect and cumulative percent did at each dose, and it explained the differences between the ED50 and the LD50, the ED50 and LD50, while the area in between called the percent required dose to achieve desired effect, the area between both of them, while to the right you can see the percent required dose for a lethal effect, the lethal effect. So the quantile dose response data provide information about the variation in sensitivity to the drug in a given population, and if the variation is small, the curve is steep. Now, what is efficacy? Efficacy, the ability of a drug to elicit a response when it interacts with a receptor. So the efficacy is dependent on the number of drug-receptor complex. Is more important than drug potency. Actually, in clinical work, we are searching and we are following the efficacy, not the potency. But when you manufacture the drug, the efficacy is very important because it means money. And a drug with a greater efficacy is more therapeutically beneficial than one that is more potent. Maximal efficacy assumes that all receptors are occupied by the drug and no increase in response will be observed if more drugs are added. This concept holds true only if there are no spare receptors. يعني ينطيني أعلى maximal efficacy، أعلى maximal effect بعد ما ينطي أعلى منه مهما ضفت drugs. While potency is another story, the concentration of a drug producing an effect that is 50% of the maximal EC50. Potency is determined mainly by the affinity of the receptor for the drug and the number of receptors available. In quantile dose response measurements, ED50, TD50, and LD50 are also potency variables, median, effective, toxic, and lethal dose respectively. Here we are talking about the amount of drugs binded to. Well, let me explain this to you. Now we have this figure. To the left, figure A, showing the drug A and drug B. In drug A, the efficacy is much more, drug A, much more efficacy than drug B, while the potency are almost same. You can see here, the potency are almost same. While in figure B, you can see still drug A and B are equally in efficacy. However, a drug A is much more potent than drug B because the amount of drug A is less than drug B binding to its receptor. Well, let's go to this figure also. You can see here drug A is more potent than drug B, but both show the same efficacy, while drug C show lower potency and lower efficacy than drug A and B. Now let's go to spare receptors. Spare receptors exist if the maximal drug response is obtained at less than 100% occupation of the receptors. If the EC50 is less than KD, spare receptors are said to be exist. See here the figure, you can see a drug effect and the KD. So you can see here the EC50 is less than the KD. That means there is a there is a spare receptors. That means not all the receptors being occupied. However, we get maximum response. Well, this might result from one of two mechanisms. That means the explanation of spare receptors. First, the duration of the activation of effector may be much greater than the duration of the drug receptor interaction. Second, the actual number of receptors may exceed the number of effector molecules available. The presence of spare receptors increases sensitivity to the agonist. These two things elucidate how the spare receptors act. Kindly beg the attention. Now, agonist, partial agonist, and inverse agonist. Receptors to have at least two states, active and inactive. يعني هو الريسبتور نفسه اكو عليه active واكو عليه inactive. Many receptor parts. Many receptor systems exhibit some activity in the absence of ligand called constitutive activity. Full agonist, a drug bind to a receptor and produce maximal biologic response that mimic the response to the endogenous ligand. هذا شو نسميه الدراج full agonist. For example, phenylephrine is an agonist at alpha-1 receptor, adrenoceptors, because it produces effect that resemble the action of the endogenous ligand norepinephrine اللي هو هذا endogenous موجود عندنا بالبودي. Now, partial agonist, a drug that binds to its receptor but produces a smaller effect at full dosage than a full agonist. Partial agonists have efficacy, intrinsic activities greater than 0 but less than that of a full agonist. In the presence of a full agonist, partial agonists act as an inhibitor. In contrast, inverse agonists have a much higher affinity for inactive state than for the active state and decrease or abolish any constitutive activity. هذا شو نسميه inverse agonist. Well, let's see here this figure. A full agonist produces complete activation of a receptor at high drug concentration in red color. You can see it. While partial agonist, the response is less. However, the inverse agonist, you can see here, inverse agonist produces a response below the baseline response measured in the absence of drug. Now, antagonist. Regarding to antagonist, a competitive and irreversible pharmacologic antagonist. If both the antagonist and the agonist bind to the same site on the receptor, they are said to be what? Competitive. The competitive antagonist will prevent an agonist from binding to its receptor and maintain the receptor in its inactive conformational state. For example, antihypertensive drug tirazosin compete with the endogenous ligand norepinephrine at alpha-1 receptor adrenoceptors, thus decreases the vascular smooth muscle tone and reduce blood pressure. While an irreversible antagonist causes a downward shift of the maximum with no shift of the curve on the dose axis unless spare receptors are present. The effect of competitive antagonist can be overcome by adding what? More agonists, while an irreversible antagonist, by contrast, cannot be overcome by adding more agonists. Competitive antagonists increase the ED50, whereas irreversible antagonists do not, unless spare receptors are produced. Well, the figure here you can see, and regarding to figure A, you can see the blue and red colors. Well, this is called competitive reversible, while here reversible, not irreversible. You can see the agonist alone, while agonist plus competitive antagonist effect of antagonist. While when we add more drugs, there's a shift of the curve, but still there's a response. While in case of agonist dose, which is irreversible, you can see agonist alone, agonist plus irreversible antagonist, the effect of antagonist what? Decrease. There are two mechanisms by which an agent can act as a non-competitive antagonist. The antagonist can bind covalently by covalent bonds or with very high affinity to the active site of the receptors, which is allosteric site, not the site of active. When it binds to this site, need to inhibit the binding of the agent to the active site, which is called irreversible antagonist. Now, B, physiologic antagonist. A physiologic antagonist binds to a different receptor molecule, producing an effect opposite to that produced by the drug it antagonizes. Thus, it differs from a pharmacologic antagonist, which interacts with the same receptor as the drug it inhibits. Familiar examples of physiologic antagonists are the antagonism of the bronchoconstrictor action of histamine by epinephrine, bronchodilator action. While in chemical antagonists, a chemical antagonist interacts directly with the drug being antagonized to remove it or to prevent it from binding to its target. Chemical antagonist does not depend on interaction with the agonist receptor, although such interaction may occur. Common examples of chemical antagonists are dimer caprol, a chelator of lead. يعني إذا صار عندنا تسمم بالليد نعطي dimer caprol, and some other toxic metals, and pyrrolidinoxime, which combine with the phosphorus in organophosphate cholinesterase inhibitor. Now, therapeutic index and therapeutic window is a measure which relates the dose of a drug required to produce a desired effect to that which produce an undesired effect. Denote the safety of the drug. يعني المقياس للسيفتي مال الدرج هو الثيرابيوتيك ويندو أو الثيرابيوتيك إندكس. Therapeutic index equals LD50 lethal dose 50 lethal dose مقسومة على ED50. The larger value of TI is the wider margin between effective dose and toxic dose. That means it is safe. For example, the drug of TI4 is more safe than that of TI2. Well, this figure showing the warfarin small therapeutic index, you can see here, in comparison to the penicillin, which is large therapeutic index. Now, signaling mechanism, the receptor effector system may be ligand-gated ion channels. That means the drug binds to a ligand with a gate, such as cholinergic nicotinic receptor. We have G protein coupled receptor مثل يعني الالفا and beta broncho adrenoreceptors, enzyme linked receptors مثل الانسولين receptor, وعندنا intracellular receptor اللي هو الستيرويد receptor. And this figure explaining in details what the previous receptor said. You can see here a ligand gated ion channel, example cholinergic nicotinic receptor. Once it binds to a distinct part in the receptor, the channels won't be opened, and sodium will enter, and this leads to change the membrane potential activity action potential and lead to give an action. While in B, you can see the G protein coupled receptor, example for alpha and beta adrenoreceptors. Once it binds to the G protein, there is activation of adenylyl cyclase, for example, and lead to protein phosphorylation. And the third one, the enzyme linked receptor, such as insulin receptor, protein and receptor phosphorylation, also lead after it binds to the enzyme. While regarding to steroid, for example, it enters inside the cell and binds to the nucleus, and with this protein and lead to protein phosphorylation and alter gene expression, and all these lead to intracellular effect. Well, transmembrane ligand-gated ion channels, the first receptor family comprises ligand-gated ion channels, are responsible for the regulation of the flow of ion across the membrane. The activity is regulated by the binding of a ligand to the channel. Response to these receptors is very rapid, few milliseconds. These receptors mediate diverse functions, including neurotransmission, cardiac conduction, and muscle contraction. One of the most examples, the stimulation of nicotinic receptor by acetylcholine, results in sodium influx, generation of an action potential, and activation of contraction in skeletal muscles. Benzodiazepine, for example, on the other hand, enhance the stimulation of the GABA aminobutyric acid receptor by GABA, resulting in increased chloride influx and hyperpolarization of the respective cells occur. Second, transmembrane G protein coupled receptor. These receptors are linked to G protein. We have G stimulatory, G inhibitory, and other types of G, having three subunits: an alpha subunit that binds guanosine triphosphate (GTP), and a beta GABA subunit. Binding of the ligand activates the G protein so that GTP is replaced GDB on the alpha subunit. Dissociation of the G protein occurs, and both the alpha GTP subunit and the beta gamma subunit subsequently interact with other cellular effector, usually an enzyme, a protein, or an ion channel. Stimulation of these receptors results in response that lasts several seconds to minutes. Well, please, my students, I want you to memorize this figure, very important, regarding to G protein. You can see here the agonist, once it binds to G protein coupled receptor, lead to dissociation of the G protein after it affecting the GDB or replace the GTP to GDB, and this lead to intra or hetero trimeric G inhibitory protein, or lead to intracellularly by increasing adenylyl cyclase intracellular recyclic AMB, and lead to alteration gene expression, cytokine production, etc., or by changing the GDB lead to phosphorylation of converting GDB to replaced by GTB, and lead to increase the recruitments of calcium from the sarcoplasmic reticulum and lead to the effect. Now we will go through the second messengers, the small molecules, metabolite or ions. Second messengers can diffuse through a cell and carry information to a wide variety of targets. Well-studied second messengers include cyclic AMP, cyclic GMP, calcium, phosphoinositidide, phosphoinositidine, and nitric oxide. All these called secondary messengers. Well, regarding to cyclic AMP is the prototypical second messenger synthesized by adenylyl cyclase under the control of many G protein coupled receptors. The activity of adenylyl cyclase can be modulated by phosphorylation and other regulatory influence. You can see here ATP converted to cyclic AMP by adenylyl cyclase, and then cyclic AMP was converted to 5-AMP by phosphodiesterase. Regarding to calcium and phosphoinositidase, phospholipase C split the phosphatidyl inositol 4,5-bisphosphate BIP2 into diacylglycerol and inositol 1,4,5-triphosphate to DAG and IP3. BIP2 will be split into DAG and IP3. DAG is limited to the membrane and activate phospholipid and calcium sensitive protein kinase C, while IP3 trigger release of calcium from the internal storage vessels in the sarcoplasmic reticulum. Regarding to enzyme link receptors, these receptors also have cytosolic enzyme activity as an integral component of their structure and function. Duration of responses to stimulation of these receptors is on the order of minutes to hours. The most common enzyme link receptors, epidermal growth factor, peptide, insulin, and others that have a tyrosine kinase activity as a part of the structure. Now, let's go through intracellular receptors. The ligand must diffuse into the cell to interact with the receptor and have liposolubility to be able to move across the target cell membrane. The activation or inactivation of these factors cause the transcription of DNA into RNA and translation of RNA into a group of proteins. For example, steroid hormone exert their action on target cell via this receptor mechanism. Now, frequent or receptor regulation, frequent or continuous exposure to agonists results in short-term diminishing of the receptor response, sometimes called tachyphylaxis. Several mechanisms are responsible for this phenomenon. Now, long-term reductions in receptor number, downregulation may occur in response to continuous exposure to agonists. The opposite change, upregulation, occur when receptor activation is blocked for a prolonged period, usually several days, by pharmacologic antagonist or by denervation.